And as all of you know, this fish is getting a lot of attention in India, but very few studies has been conducted on its behavior till now. So what I will do that in the initial segment, I will show you some of the works which we are doing, keeping the behavior of the fish in the focus. Parallelly, we have another line of work also is going on where we keep the human behavior and we try to understand how the human behavioral element can be utilized for the conservation. So that is the plan for today. But the second part, human part, I am not speaking today. So the talk is titled, as you can see, Understanding the Tigers of the River, Behavioral Insights to Support Conservation of the Freshwater Megafishes, mahseers. So mahseers are also known as megafishes because they many of them grow to huge size. So you can see mahseer is not a single species; it's a group of fishes, and many of them comes grow up to 30 kilogram in size. And many species are keystone species in nature, and they are very well renowned game fishes. And even in the Indus Valley seals, people have seen the etching of mahseers. People has made the figures of mahseers in that. And it's called as God's fish also in many regions because many communities worship them as and many offer them food, many offer them puja, and consider them as holy in nature. You can see the size of Himalayan mahseers got. So generally, mahseer comes under three genera. One is Tor, second is Neolysocylus, third is Nasitor. And in India, we have around 22 species. Out of the 56 known species, 22 can be seen in India. And their large size and the top positions occupied in the food chain make them vulnerable to any changes happening in the environment. And in many contexts, what happened that people say that they may be the easy victims of climate change in future because the demand of the food required is very high. So is the other demands. So many people suggest we should start thinking about a project Mercy in the line of Project Tiger so that we may be able to protect these fishes. It is, as I told you earlier, it is one of the most popular game fish in India. And since 19th century, people from different parts of the world came to India to angle mahseers. Now also it is happening. And for many states, this fish provides a lot of revenue through ecotourism and angling. So this is economically and ecologically important for India. But many species of mahseers are facing decline even though they are angled, even though they are worshipped, even though people consider them as very important, their populations are declining in many areas. People believes habitat degradation is one of the reason, and river fragmentation, dams are causing the problems for fishes, and absence of larger conservation community and coordination of the activities between different departments are also not seen in many areas. An illegal and unsuitable fishing method used by the people also reduces this fish since its size is very large. People target them. And a lack of awareness among the local communities, even though they know that the mahseer is there, but many of them consider it as a delicacy, especially in different states of India. And many contexts, what happens that the law to protect these fishes are implemented with a lot of loopholes in that. So many contexts, judiciary or executives suggested that we need to do something to protect these fishes. So these are some of the conservation attempts done in India. Whatever you are seeing as black spots in the photograph, this photo is taken by one of my PhD student, Prandik Das. You can see that the fishes, these black spots you are seeing, the are fishes, and it's a temple sanctuary where people go there, feed them, and the fishes gets aggregated there. And another major intervention for conservation done here is reintroduction and restocking. So artificial propagation has been standardized for selected species like Tor putitora, Tor kudri, and many organizations every year release large number of young ones into the natural water bodies. So these young ones are reared in artificial environments like hatcheries and transported to different parts of India and released in the natural habitats like dams, rivers. So one of the major producer of seed in India is Tata Power Company Limited, it's Lonavala. So a article published by them shows that from 1970 to 1994, 62 lakh young ones has already been produced and released, around 2 lakh young ones per year. And another hatchery located in Karnataka, where my institute and my institute is located, and from where I am getting the young ones for my experiments, started in 1992, and they are also producing 25,000 to 35,000 young ones a year. And recent exploration, some published data conducted by my team shows that in India, 13 hatcheries in different states produces around 4 to 5 lakhs young ones every year. And these are also released into the natural water body. So, but one of the major problem is many species, you don't see even after this much of efforts, many species didn't many species or many populations didn't get the original size back. And exploration shows that rate of success is very low for this kind of released young ones. And there is another interesting thing to say. There was no systematic exploration or data available about the 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 fate of the young one who has already been released into the natural water bodies. So, but the studies from Europe and America shows that the megafishes generally, when they're released from the hatchery, only 3 to 5% survive because they don't have the behavior profile required to survive in the natural habitat. So, Masif, even though this is not studied, we also think that this may be one of the reason they also may be facing a lot of behavioral deficiencies when they are generated and produced en masse in the hatcheries. So the studies has already shown that the homogeneous environments of the hatchery, not having any selection pressure, in many contexts fail these fishes to develop the behavioral profile required to survive in the natural habitat. And as you know, the most of these natural habitats in the tropical region like India, like Western Ghats, which is a biodiversity hotspot, the number of predators and the complexity of the environment is also very high. So animals may not get, the released fishes may not get enough time to develop to fill the gaps in their behavioral profile, such as foraging behavior, social behavior, anti-predatory behavior, reproductive behavior, etc. And in many contexts, their development of their neural structure also will be different because of the homogeneous environment in which they survive. In this context, we suggest that if we want to come out with a model for mahseer conservation, we need to have the data of ecology from different sides of ecology of the fishes. We need to have the behavioral data of the fish, and we need to have the behavior of the people or different stakeholders interact with them. So you can see here, ecology is being studied by many groups in India, and behavior of the people data. We let us see what is the scenario. So in the initial phase of our study, before starting our experiment part, what we did that we explored the publication related to behavior of the mahseer, which we are going to focus in the current talk. So yeah, so this is what I have already shown in the previous slide. So here you can see we searched the existing databases to know the papers related containing the term behavior and mahseer. And we searched most of the prominent search engines like Google Scholar, Scopus, Science Direct, and all that. And we considered the a 15 years period starting from 2007 to 2022. We have the recent data also with us, which shows mild changes in the last 2 years. And what we found was that you can see the trend of papers coming on mahseer behavior research in this slide. So only 42 to 43 papers containing the term behavior was available. And in many contexts, what happens that that was not completely covered. Just it may be a mention of some behaviors like fish was moving like that. They selected this food particles, this food items like that. And the most of the papers were focusing on one species, that is golden mahseer. So majority of the papers, around 39 papers of this 43, was focusing only on one species, that is golden mahseer, even though we have around 22 species in India. And most of the behaviors focused was mainly on, like you can see in this slide, that it was mainly on the behavior, they like the foraging and migration like that. So very little information on the behavior is available in the market. So then keeping this scenario and with an aim to fill this gap, we started our exploration keeping Tor Kudri's young ones as the model. Because like I told you earlier, this is also known as bluefin mahseer and Deccan mahseer, which is common in the Kaveri River basin in India and produced and restocked by agencies, government and non-governmental agencies in South India. And many contexts, like I said, they also grow to large size, angled by many people. And we try to understand what are the factors affecting their behavior or what is the behavior strategy they use in different contexts and all that. So the problem when we started this research was that there was no data on the behavior of the torcudri available, even in the environmental, even in the laboratory setups. So keeping this in scenario in the mind, what we did that we started to explore, we decided to study the behavior and cognitive abilities of the torcudri young ones. So we were mainly focusing the 2-month-old young ones. We get them from the hatchery, which are actually procured, produced, and produced for releasing in different parts of Karnataka and the South Indian natural habitats by Karnataka Fisheries Department. And they were very generous towards us, and they provided the young ones whenever we asked them. And their hatchery, located in Harangi, Coorg, is where they do the artificial propagation and generate these young ones. So we procured the young ones through the fisheries department, and we started to study the personality trait and behavioral syndrome in this fish. Because as all of you know, the behavioral flexibility will become one of a major factor playing the survival rate or deciding the survival rate in fishes when they are released into the natural environment. And there is a plethora of literature available on other species talking about the importance of personality traits and behavioral syndrome and how it can affect the desired changes in the behavior, which can help the fish to survive in the natural environment post-release. So, but as I told you, this here we started with, since there was no information on this was available with mahseer, so we need to generate the base. We were compelled to generate all the baseline data also for mahseer so that we can continue our exploration. So we focused mainly three personality traits. One is boldness. That is a propensity to take risk. I know most of the animal behaviorists attending this talk are familiar with these terms because it's now a very prominent term in the field of animal behavior. And another term, another activity we considered. And the third element we considered was sociability. So at that time, Dr. Vishnath Verma joined my lab as a postdoctoral fellow, DBT postdoctoral fellow, and we decided to start this exploration with some of our other friends. So, uh, as like I said in the previous cases, these personality traits are often linked to each other, and in many contexts, this is for the youngsters who is attending this talk, are tightly linked to each other. And in many contexts, that tight linkage between these behavioral traits limits the individual's ability to cope with the environment. So if we don't know how this kind of bridging and connection and disconnection of this behavior happening, and with how these behaviors are linked together, it will be difficult for us to understand or make any intervention to reduce the to reduce this the post-release mortality. And in many contexts, what happened that if the behavioral activity syndrome, which is studied in many species, shows that if the high predator pressure environment, such a syndrome will work negatively towards this individual. So similarly, sociability, because they, as I told you, the young ones of mahseer are very social in nature. They move more like a tight shawl, and you can see they're moving whenever they are young. They are in the aquarium. They move as groups and do the activities in groups. So we need to understand how the sociability is also working here, or sociability is also influencing their behavior. So, but when we talk about sociability syndrome, you may know that it is affected by the early environment, and it is species specific. But the sociability boldness syndrome also affects the structure and dynamics of the population. We will see that some of the glimpses in mahseer in the coming slides. So we need to understand the behavioral syndrome. So we decided to explore the behavioral syndrome in the species. So the apparatus used is swimway. And as you know, swimway apparatus is like this. Here there is a height or a chamber, small chamber, where what we do that we release the fish here in this chamber, and then we give them time. And after some time, what happens that the fish, whoever is bold, will come out fast, and the shy fishes will take more time to move from here to this chamber. So what we did that we studied the boldness of boldness activity syndrome using this apparatus. So the fishes were test fishes were introduced here, and we saw how long they will be taking it to come out of it, and how they spend their time in this chamber. And the dimension of the apparatus you can get here, and what are the parameters we use, that also is clearly given in this. So we were given 5 minutes for them, and we were given a ceiling value of 300 seconds for this fish. If they don't come out of this chamber after 300 seconds, what we will do that we will stop the trial. So we tested 61 individuals like that. And like I said in the previous slide, boldness is here defined as the time taken by the fish to come out of the start chamber. And another parameter considered was visiting the medial region of the swimway apparatus. Number of times the fish swims in a chamber in an area at distance of at least 5 centimeters from the closest edge within chamber B. And the other parameter we checked was activity, the time spent actively moving in the swimway, frequency of switching between the chamber A and B. And the videos were recorded. Videos of this experiment were recorded. And it was analyzed using the video analysis software Boris. So what was the result we got? We found that in this species, bolder individuals are very active. So not much new result, because in many other species also people has proven that bold individuals are very active. And here also we found that they are very active. And they visit, and generally what they do that this is the relationship you can see, where switching between the chambers and boldness, and switching between the visiting the medial region and boldness here. And here you can see how the activity is correlated with switching between the chamber and the number of times they crosses the medial area of active fishes. So this is the analysis part. Here what we did that we went for a zero inflated Poisson regression, because there was more cases. There are many cases where the fish didn't come out of that. This exploratory type as responsible variables, and boldness and activity as the fixed variables. We tested this. And what are the insights we got from this study? Bold juvenile Deccan mahseer show greater activity and visit medial region of the apparatus more frequently switch between the chambers. So active risk-taking individuals explore all areas available in the habitat in a habitat. So interestingly, you can see that this happens in isolation. So we found that if you release an individual in isolation, it will be exploring this area more if its personality is bold. So at the same time, the problem is that in most of the natural environments, the individuals are not going to be released alone. So we will be either releasing them in huge numbers, like you have seen in the previous slides, or with the other species also. Because in many contexts, what happened that in the same habitat, they release the fishes with the mahseer with the species of economics importance. That we can see in the coming slides. So then what we did that we tried to understand the relationship between sociability and boldness. And along with that, we kept another parameter that is laterality. Because, fishes, it is easy to study the lateral utilization of the brain because there is no isolateral connection and the connections are mainly contralateral in nature. So what will happen that fish has to change its position of the body to see the stimulus nearby. So we want to know because we try to keep two elements here. One is sociability. Sociability with not only the conspecific but it with alien invasive species because many recent studies shows that the alien invasive species tilapia, common carp are common in the habitat of uh, Deccan Mercier and the places where they are released. So in many cases, their populations are also increasing. And in many contexts, many organizations also release this fish because it provides livelihood for the local population. So what we did that we try to understand how the sociability exhibited towards alien invasive species, tilapia, and another member of its own species are related with each other. So the study, what we did that we continued the exploration in the same line. And this was the apparatus used for that study. Here also, here you can see in this apparatus, chamber A is the release chamber and B is a channel through which the fish has to move to reach the channel, the chamber C, which contains a chamber where subject fish is kept. So here what you can see that here, the subject fish will be introduced and they will, after some time, they will slowly move here and those who are not moving will be moved with the support of a scale and they will come here and then they will explore the stimulus here. And many studies are available to show how this apparatus is being utilized by many others earlier. And we kept three different stimuli here. One is unfamiliar conspecific, familiar conspecific, and empty cage as the control. And then what we did that we tested the 61 individuals and we studied the time spent by the focal fish within 2 centimeters of the stimulus cage. So what happened that the time spent to this is a sign of sociability. And what you can see that either the fish will move clockwise or anti-clockwise, keeping the stimulus in the center. So from which we can calculate the laterality index using this formula. That is duration using left eye minus duration using right eye divided by the duration using left eye plus uh, right eye. So from this, we will get how each individual's laterality. So what we did that before this fishes were subjected to this analysis, they were tested for their boldness and activity using the previous apparatus I mentioned in the previous slide. So here is the result. What you can see that here you can see in the control condition versus the stimulus when it is conspecific and heterospecific. Here on the y-axis, you can see the time spent near stimulus. Here you can see it spent more time with the conspecific. And the heterospecific was not that much preferred. So it shows a very high species specific preference pattern here when there are two individuals are available as a stimulus. And when we compared, when me and Vishnu compared the total time spent near stimulus versus the time to emerge from the shelter, that is boldness, what we found was that it is linked. These traits are connected. And more bold Mahseer spends more time near the. So from this result, we can understand that the bolder individuals spend more time near their conspecific than heterospecific. And like you can see, those who are interested more in the analysis part, they can see it here. Uh, what happens that the boldness was again the individual shows consistent difference in the time spent near the stimulus chamber. So that you can see from this study. So what this results point towards that, so bold individuals, boldness and sociability is correlated in the species, and bold Mercier prefer to spend more time with their conspecific. So we checked the laterality. What is, because in many contexts what happens that lateralization helps the animal to take decision quickly in a changing environment. And as you know, they are generally released into dams and rivers. And during months, sometime they may be released in monsoon time. At that time, the environment also changes quickly. Not only that, they will have to take a lot of decisions because the environment is too complex in comparison to the environment in which they were born and brought up. So here you can see when we check for the laterality in the control case versus stimulus that is conspecific with heterospecific, you didn't get much difference here. So laterality was not an important thing, important factor here, or they were not lateralized. Their behavior, there was, we didn't get much any bias towards the stimulus when they were using their brain. So the time to emerge from the shelter was, when we compared that also, you can see it was also not much significantly different. So showing that laterality don't have much role to play in these cases. So what is the insight we got from this result? So this result revealed, showed us that so the preference for so there is a preference for conspecific over heterospecific in a decision-making scenario. And bold individuals are more likely to be sociable towards their own species than alluvial invasive species. And no significant difference in the lateralization exhibited towards the different social stimulus came, and it was not consistent with an individual across different stimulus social situation, and it was not correlated with the personality trait. That is the inference we get from that. So we can say that bold individuals prefer to spend more time with their own species, and they are more social. They may spend more time with their species, and lateralization is not connected. So generally, this has a if as a conservation importance, because this result has a conservation importance, because many studies have shown that bold fishes are the one when you release or when they move to a new environment, when a colony or a group is moving to a new environment, bold fishes are the one who are going to give and establish new colonies or move and spread into the environment. Here you can see that boldness was tightly linked with the sociability, and bold fishes were staying there more with the groups. So we need to test this situation in the natural environment to see how the spreading affects in a and how the personality of the individuals in the group may be affecting the spreading of that individuals into the newer areas. And many contexts, even since they have sociability is high, so they may try to join alien invasive species also in the absence of conspecific. Because you can see, as you saw in this experiment, because there was preference, even though it was not significantly different than the conspecific. And in many contexts, living with alien invasive species can have many undesirable effects. And absence of lateralization, that is which we saw here in social context, is linked with many cognitive deficiencies in many species which need to be further inquired in the species, because how it is going to affect their survival in the field, in the natural environment, like that. So but there are some drawbacks also for our exploration. The individual tested in this study are only 2 months old, and Malseers live for long, and we don't have any data till now to compare with the laboratory and environment, the hatchery riot and natural population. So this interaction between hatchery riot and invasive species also need to be studied in detail. So that we will see in the coming slides what we are doing nowadays to understand that. So then, if that is the situation, you found that yeah, the older individuals are active, but in a natural environment, in a when they are in isolation, and what you found was that the personality traits bold individuals are interested to join with their own species, and we found this. So then we need to, what we were planning to, we test try to prove it in the decision making scenario. So here it is only one stimulus available where they are showing this behavior. What will happen if that is put into a decision making scenario? Last point we discussed also, we said that laterality was not there, and many studies shows that it can affect decision making in fishes and all that. So we try to understand the shoaling, keeping shoaling behavior as a criteria. We try to understand the decision making in the mahseer, juvenile Deccan mahseer. And as all you, as many of you know, joining a shawl is very much important, and group life helps them, provides them a lot of benefit over a small cost. And what we found was that the species, the body size, number, sex, parasites, disease, competitive ability, a large number of parameters has been tested, how it is going to affect the decision making in different species of fishes. But Mahseer, no information is available till now, especially in Torkudri, the species which we are studying. So we try to understand, and there are many studies shows that shoaling decisions are a result of trade-off between complex interaction between these factors. It's not one factor affecting at a time like that. So we try to explore how personality affects of shoaling decisions here, and influence of as you know, there are many studies in this line. Studies showed that shy European sea bass and zebrafish show stronger preference for familiar individuals over unfamiliar ones. Uh, because the aversive nature of shy individuals could avoid novel situation and the heightened level of conflict associated with unfamiliar individuals. And less sociable guppies show stronger preference for larger shoals. And less sociable individuals are also shown to switch more often between the shoals. So here, less in our case, it will be like less sociable means it maybe they are also show between the chambers. So we need to know how this in many how this the stalling decisions are how we were trying to understand the stalling decision making in the juvenile Deccan Mercy. So what we did that this is already known fact. Sometime what happened that if there is a alien invasive species is associated, that also can affect the decision making scenario. So keeping these factors in the mind, what we did that we tried to ask these questions: how juvenile Deccan mercies would prefer larger shoals over smaller shoals, or familiar individuals or unfamiliar ones, members of their own species over non-native species in decision-making scenarios. So we added one layer of complexity to the previous set of experiment. We brought the idea of decision-making here. And we again try to understand whether the strength of this preference would be correlated with personality traits, that is boldness and sociability in this species. So keeping this idea in the mind, we try to test this social decision-making experiments. As you know, social decision-making experiments are generally done in this kind of apparatus, and these experiments are called titration experiments by Ian Barber et al. When many other authors are calling it the titration experiments. So what we did that here, what we do that the stimulus shoals are given on the sides, and the test individuals are kept in this presentation cage, which is liftable. So here we will give the stimulus. Here also another stimulus, and then 5 minutes will be given for acclimation after releasing the test fish here. Here also we use the individuals, not the groups. And the test time is 10 minutes, and this is the preference zone, and we will see how much time they spent in this. So what were the parameters tested? On one side we kept mahseer, unfamiliar mahseer, other side familiar mahseer, and another combination was unfamiliar mahseer versus unfamiliar tilapia, familiar mahseer versus unfamiliar tilapia individuals. And we don't have any information how long they take to develop familiarity. So what we did that we kept a group of fishes for around 8 months together, so that it can lead to whatever, it's a long duration for developing familiarity. So my previous work using another species of fish called climbing perch shows that within 14 to 20 days they will generate familiarity with the other species, but here we don't have any data like that. So we went for the extreme uh, exposure this time, that is 8 months, and each subjects were exposed to all three treatments, and 8 days were given as a gap between the tests, so that the previous experiment does not affect the fish. So here mainly we use the parameter, that is preference index, the time spent along familiar shoal divided by total time spent along both shoals, and we checked the significant difference from the chance in this context using one sample t-test against a value of 0.5. And this was the result we got. There was no significant difference from the chance, like you can see here. And we conducted LLM over that, and no significant effect of boldness also was found here. Here you can see the slide showing the familiarity and conspecific preferences here. This is a situation of unfamiliar conspecific with heterospecific, familiar conspecific with versus unfamiliar conspecific, familiar conspecific versus unfamiliar heterospecific. This is a scenario, and you can see not much variation in the preference shown to any of the species, any of the stimulus here. And here you can see what the result of our test conducted to understand the relationship between boldness and the shoaling preferences. So this is the statistical part for those who are interested. That. So then, when we tried for the post-hoc comparison using Tukey's HST test, it is revealed that there was not much. It was revealed that there was not much difference. And no consistency was observed in the preference showed by the individual fish for familiar individuals over unfamiliar individuals, or the conspecific over heterospecific. So that you can see in the previous class. And no shoaling preference based on the familiarity of species when stimulus are individual, that also we found here. And shoaling decisions do not appear to be consistent amongst the individual fish, or related to, or in bracket related to their boldness. That is a personality trait. So the thing is that many contexts, what insights we get from this is that many species join heterospecifics to avoid oddity effect. And mahseer shows no preference towards the familiar conspecific over unfamiliar conspecific, and it failed to exhibit preference for an isolated member of its own species when presented in combination with a non-native heterospecific tilapia. So sometimes we feel that when we did a visual analysis of the features for a human eye, in many contexts, the young tilapia and young mahseer, maybe there are very minimal visual difference may be seen when they are very young. But when they grows, they change the shape a little, and they evolve, they develop into the individuals with the difference in their morphology. So other hypothesis need to be tested in this context is maybe this species may be incapable of acquiring individual familiarity with conspecific and heterospecific, maybe because of their captive life, where they never get any chance to get any other species. So that need to be tested to get a clear picture of the behavior of the mahseer. And and this also need to be studied, because there are studies which shows that this preferences changes according to the natural habitat, and with the age also, and the context also. So then what happens that there are studies which shows that 50 days old guppies demonstrated discrimination of shoals based on group size and body size, while 10 days guppies were not able to do that. So considering the age or the longevity, or the lifespan of the mahseer, we need to generate such a data if you want to tell something specifically about these results. So we continued the exploration in this line, so that what happened that uh, so that we have two uh, we went for a shawl choice experiment. So the situation was that we went for main things, that is 2 versus 2, 2 versus 3, 2 versus 4, 2 versus 8 shawls. And we kept a 0 versus 2 also as to also because it's more like a control, or it can work as a sociability. So what we found was that there was 8 days rest between successive trials here also, so that there is no issue of this. So what we did was that the apparatus was same. On one side we gave the stimuluses like this, and other side we gave the other stimuluses like that, and the n was 34 in this case. So the preference index was taken like in the previous cases, and the linear mixed model was conducted, keeping treatments as the variables. And we used AAC, delta AAC and took his Horner's significant test. And what we did was that a one sample test of PI values against 0.5 was also conducted. When we conducted this, we found that in 0 versus 2, repeating or supporting our previous result, what we found was that these fishes preferred to stay with the groups with a shawl over an empty chamber in a decision-making scenario. And the preference index, we tested another point, that is preference index, how it is correlated with the latency to first approach the shoal. And that also was not significantly correlated. So we found that in the scenario 2 versus 4, 2 versus 8, there was significant difference. And in other cases, there was no significant preferences shown by the species. So generally what happened that they moved to the large shoals when in 2 is to 2 situation and for 2 is to 3 situation, there was no clear-cut preference came, but when it moved forward, it started showing more and more preferences towards the larger shoals. And here you can see, here you clearly you can see that the shoaling preference had in much connection with the personality traits also here. So the personality trait was not correlated with the shoaling preference here. So the shawl size preference, this is the statistics of the shawl size preference, as you can see here. And this is the data we got from the linear mixed modeling. And you can see that larger preference for larger shawls is apparently only at 2 versus 4 and 2 versus 8, and is not related to boldness or sociability in mahseers. So that was a very interesting finding. So then again, they may prefer the larger shawls if they see there's a chance that when you release them, they may go for a larger shawl because the shawl in a natural environment, they may be getting multiple shawls and they may be going for a larger shawls. And sometime what happened that there may be the shawls may be that of alien invasive species or other things, other species like predator species like that. And other alternate argument comes here is that in this species, numerical acuity is low. You can see that the ratio 0.5 2 versus 4 than in guppies and three-spined stickleback, which people has already discovered earlier. Alternatively, this fish may be capable of, another hypothesis need to be tested is they may be capable of discriminating between 2 versus 3, but the benefit of shoaling with 3 fishes compared to 2 may not be sufficient to elicit a significant preference of the large shoal. So that are the other hypotheses we are testing in the next set of experiments. Because here it is not clear what could be the reason they go like that. So then how long? How much more time I have? Tarun?
Dr.
Hello?
Binoy, you have around 10 more minutes.
Okay, good. So I will finish that. So I will tell you about one more experiment which is continuing now. Then I can stop. So what happened that we tested we tested the personality traits, we tested this and all that. Right? So then we try to see the another element, that is how personality. So there are studies which shows that personality traits can affect many cognitive abilities, including learning and personality expression. And there are some recent studies which shows that personality expression is contingent on social context. And as you know, we are not going to get a single personality group. The group will be a group of mix of animals. And group decision requires agreement between different personalities in many contexts. So what we did that, in order to explore this scenario, what we did that we started testing the pairs differing in their personality traits. So what we did that we studied the pairs, the pair and individuals in spatial learning scenarios. And we even did an experiment keeping the alien invasive species uh, also involving alien invasive species. So the first case, it was individuals difference in their personality traits, that is bold and shy individuals. We separated bold and shy individuals using the apparatus which I have shown earlier. And bold individuals are the individuals which crosses the which comes to the chamber B uh, before 200 seconds. Others, shy individuals generally don't come even after taking 10 minutes to the chamber. And we made bold. Then what we did that we tested the individuals. Then we made bold pairs and shy pairs and pairs of common carp and mahseer were both individuals were shy. So we did this experiment to see how these pairs perform in a learning scenario, because that's also contributed to the situation, because these animals will have to learn new things in group as well as individual when they are released to the natural environment. So this is a spatial maze we used. This is the release chamber. There is a gate one, and this is the chamber where if it reaches to this chamber, this will be like a wrong chamber. It will have to come back again to reach the other chamber. It's like a blind chamber. And on the end, we provide a group of conspecifics that are the reward. So here we used other individuals as the reward, and we repeatedly tested the behavior of these fishes, maybe individual or group in this scenario for 10 days. So every day they will be getting 10 minutes, and we studied their behavior, different behaviors that I'm only showing, considering the timing, I'm only showing the learning part. So what we found was that you can see here, this left side data is a data of bold individual, right side is the shy individual at individual level in this maze. You can see day one up to day 10, you can see around up to day 5, around 60% of the individuals started reaching the reward chamber. By day 10, around 70 to 80%, around 70% of them, more than 70% of them started to reach there. But shy individuals, this was a scenario, because by the 10th day also, only 20% of the individual reached there. And when we tested them in pairs, we got a very interesting result. You can see the day 10 scenario in the bold pairs. Initially, they were actually, in the previous cases, you saw how the bold individual reached this last chamber by day 10. Here you can see none of them reached that. So when they behave as individuals and when they behave as pairs, the personality traits matters here. So the shy pair, you can see the scenario, not much different. And when we made the pairs of heterospecific with a heterospecific, it was not much different from the previous case. Around below 20% of them reached there in that scenario. So that was the situation we found here. That so then the other thing, what insight we are getting here is bold individuals learn fast in isolation compared to their shy counterparts. And when they are paired with another bold individual, they don't reach the reward chamber. Okay, so bold individual learns fast in isolation, but when they move with another individual, when they are introduced into the same situation with another individual, they don't reach the reward chamber easily. So the shy individuals behaved similarly in the presence of conspecific and heterospecific in this learning scenario. So the result shows that personality traits of the members can affect learning by the payers, at least in payers. And we need to test what will happen if the number of the individual increases, the number of the group size increases. So considering the time, I'm concluding the works which is going on, which I'm presenting here. So Boulder, Jonel, Deccan, Masir, as you show, as you seen, grows higher activity in isolation, and they were not the familiarity was not a factor when they were tested in the decision-making scenario. And bold individuals are more likely to be sociable towards their own species than alien invasive species. We saw that in the previous slides. And there was not much significant lateralization exhibited towards different social stimulus. Hatchery-reared juvenile Deccan mercies prefer to join shocks with greater number of conspecifics. Uh, because, as you have seen here, they prefer to join the conspecifics. And bold individuals learn fast in isolation compared to their shy counterparts, but not pairs. And shy individuals behaved similarly in presence of conspecific and heterospecific in learning scenario. So even though we have this result, one of the limitations which we see is very few behavioral data is available on any massive species. We need to generate such information if we want to come out with hatchery training protocol or life skill training protocol or any behavior-based intervention for the species. Because as you have seen now, very few works has been already done, and we also did this much only. And another lacuna here is we don't have much information on the fishes collected from the natural environment, which we have started recently collecting fishes from the natural environment and identifying their age and all that, and to try to compare it with the natural the species, the individual born and brought up in the hatchery situation. So we hope that we may be able to come out with more comprehensive picture of the behavior of mercies, and along with the data we are generating on the other side, that is the human behavior data, we may be able to give a pictures for the policymakers so that the mercy conservation programs are benefited. So this is the last slide. I thank these people for their support. And these projects were funded by these organizations. So thank you very much.
Thank you, Dr.
I
Binoy.
hope I finished on time.
Yes. We have some time for the questions.
Yes.
Before we move to the questions, we had some technical issue at the beginning of the session. So I'd like to again welcome everybody to the first session of the ABL Behavior Live Conference 2025. And I'm Tarun. I'm chairing today's first session, and we had just heard Dr. Binoy talk about his work. Dr. Binoy is an assistant professor at the National Institute of Advanced Studies in Bangalore, India. And as we have listened to his talk, his work, their lab's work integrates lots of behavioral insights from the lab environment, and then integrates that together with what's happening in the natural environment for a better conservation strategies of the mercy fish. And I would like to request everybody, every participant, to direct your questions over at the YouTube live chat. So Dr. Binoy, we have few questions. So David says, thank you for a fascinating talk. How do you think that the conservation methods can be changed to be more successful?
See, one of the thing is generally we give more emphasis to biology-based interventions, because when we talk about the conservation, the most important element coming to our mind is how to increase the number of individuals. But at the same time, now you can see even in a population, the individuals behave differently. And the many contexts, what happened that we don't integrate the behavioral data in this conservation element. So we need to understand where, like you saw in the initial slides, what is the data available on the behavior of the mouse here? Even though it's a charismatic species. The most interesting element is it's a charismatic species. It's not like the species I was working earlier, that is climbing perch. A very small fish has economical value, but not much popular. So this is a charismatic species. Many groups use it as an important species, important icon. It's icon, and governments are also investing on this, but we don't have the behavioral data. So we need to think about two behaviors. One is the behavior of the animal, focal animal, and the flexibility that can bring in the focal animal's behavior. And second thing, the most important species which have the capability to change the environment, that is humans. So that is a scenario because we need to consider more behavior data, and we need to understand how the animals are going to work in different environmental conditions and how they are going to adapt their behavior. What are the ecological, what are the evolutionary traps to which they may fall? Then only we may be able to come out with more effective conservation. Otherwise, like you saw, this much number of young ones are released into the natural environment.
Yeah.
Not only in this study, many other studies also have shown that if you don't know where is the behavioral lacunae, we will not be able to help the animals. So
Okay.
is the other stakeholders working on that animal.
Okay. So next we have a question from Catherine. Catherine, thank you for the talk. Do you think the shoal size preference has a maximum number of individuals, a ceiling effect at 10 or 12 individuals, for example?
In my studies, these studies I didn't consider shoal size because the shoal size was small, up to 8 only we went. So people who explored larger shoals found that they after generally the animals use Raoult's law. So many of them says that the number may not matter, the area covered will be used at that time while taking a decision. See, because there should be a trade-off between the mechanism used to take a choice, correct? On one side you have a small shoal, on the other side you have a larger shoal. We don't know how the brain converts it as a neural currency and compare it that. But after a large shoal size, about 10 and all that, in this species we don't know. We didn't test that because we went up to 8 only. Above that we don't know what is the situation. So otherwise, so generally what happened that they go for this kind of thing. And when two large shoals comes, the area covered, the density of the individuals, that kind of things will be used by the field. That is what the literature shows. But in my species, we tested up to this much only.
Okay. We have another question from Leah. For the experiment on the sociality, did you look at the behavior of the fish in tank D and whether it could influence the behavior of the focal fish?
No. So sorry, that is a very interesting and important question. We often says that often there are people says that because if there is an interaction, for instance, in one side, if there is a fish which this fish has some connection, it can generate some connection, or if that their behavior, like in communication, the way the other agent interacts matters. So we didn't check that yet, but we have the videos. We have the videos of that. So we can do that in future, but I don't. In this kind of studies, we focus only on the stimulus fish, considering the complexity of the data.
Okay. We have another question from Tang. Do you know how climate change alters mahseer behavior and their habitat use?
See, again, we don't have much data on that, but we expect, like I said in my initial slide, these are huge fishes. They grow up to large size in their body, and so their biological demands are also very high. And many of them, not the species I am working on, but the sister species are living in Himalayan waters. And many of them breed in cold waters. So temperature change can affect them a lot.
Yeah.
And some studies recently, some of the people in India itself have started exploring what will happen if the wavelength, the daylight, dark light cycle changes and all that. They have started exploring that now. Okay. Because people expect this fish may be one of the earlier victims of this kind of changes happening in that.
Okay. So I would like to ask one question. So
carry on.
Since you're also the in charge of the science communication program, so and you have worked extensively now on the behavioral aspects of the mahseer, and one thing that you mentioned when this restocking program was done was the success rate was very low, and then there were if I'm not wrong, there were like 13 hatcheries in India who have been releasing them.
Yeah.
So have you been working on the science communication aspect to relay the results from your lab to them?
Yes, because one of my PhD student, Prantik Das, who is planning to submit soon, has conducted extensive field-based studies on stakeholders' perception, starting from hatchery people and how the stakeholders from different species, working on different species of mahseer from different states. And he's now writing his thesis, that part. So once that is ready, we are planning to contact policymakers, showing that where we can pitch in so that our fishes are not lost.
Okay. Thank you, Dr. Binoy, for your exciting talk.
Thank you.